Okay, so welcome again to our fourth Meet the Experts seminar in the Meet the Experts seminar series. I want to take just 1 minute before I introduce Dr. Guo to just direct your attention to the website for, sorry about that, the website for the Centers Centers of Excellence in Hematology. So I'm sharing my screen here. This is the website. It's very easy to get to, which will show you up and coming speaker events, and it shows you opportunities for funding. It also shows you some of the cores highlighted. And if you scroll across the bar here, you can find other centers, all the different centers that are supported by this mechanism. And one of the things I really want to hone in on is this opportunity right here, which is the funding opportunities that are provided by the Centers of Excellence in Hematology, where they support you applying for what are called Type A and Type B grants. The Type A grants can be applied for at any time, and they're available monies that can be sent to a core facility that you want to use for your research. And so you can apply for those at any time. And the application process is down here at the bottom of the page, and instructions follow here. The Type B grants calls are made about twice a year. We've just recently done a call and received applications and are just in the process of announcing those awards. And those will have another call go out probably in July, I believe. So I highly encourage you to go to this website to figure out what the opportunities are here, but also to learn about what the different centers of excellence, different institutes have as cores that could really advance your research. So that's my little pitch on that part. Then I want to introduce our speaker for today. And so this is the flyer for that hopefully you all got. And so I'm going to mispronounce your name, and I should have asked that at the beginning too. Tell me how to pronounce your last name.
Guo.
Ha. See, I need to speak Chinese. I'm telling you, I'm so frustrated by my inability to pronounce names. But anyway.
No worries. No worries.
Okay. So today we're going to have Dr. Guo speak to us. She comes from Yale. And she received her bachelor's degree in China and then went on to a Ph.D. at Boston University, where she worked in the laboratory of Yale Shangqin, studying HER2 new signaling. She then moved on to a postdoctoral fellowship at Harvard and Mass General in the laboratory of David Shaden, where she studied microRNAs and microRNA regulation of gene expression. She established her own research laboratory in 2013 at Yale, and has been working on stem cells, cell cycle, and stem cell hematopoietic lineage. And today she's going to talk to us, and the title of her talk is Reading Live Cell Cycle Speed by Fluorescence. And I'm going to unshare and now let you share.
Okay. Can everyone see it? See my slides? Can you see that? Okay.
See, yep, there you go. Perfect.
All righty. Thank you so much, Diane, for organizing this fabulous series. And thank you for everyone to join in. If you have questions during the talk, feel free to interrupt me. Anyway, you would like. As you see, the title I gave Diane a few months ago is Reading Live Cell Cycle Speed by Fluorescence. I want to emphasize that my goal here today, or my mission to accomplish really, is to really represent and to promote the Yale CCEH, particularly imaging core. Myself and my colleague, Jörg Biersdorf, we co-directed this imaging core with the single mission that we want to accomplish is to make imaging more widely accessible to all of you. So how do we do that? I will spend today on two kind of topics and to demonstrate how our imaging core basically try to democratize some of this imaging technologies so that everyone can use it. So let me start off by saying that depending on your research question and the cells or tissues that you're about to study, there's a large variety of imaging techniques, really for your question. So deep in the tissue, there's this 2-photon microscopy that can penetrate, for example, in the bone marrow studies, in the calvarium, or in the past several years, we can see also deep tissue. After tissue clearing, we can see the cells inside. And depending on the resolution, we can also resolve, across the light diffraction limit to see subcellular or ultrastructural microscopy that usually is driven by the super resolution microscopy. And for the longer term imaging that over generations for days onward, there's this long-term live cell imaging and tracking. For most of these techniques, imaging techniques, it either requires sophisticated high-end imaging equipment or the technical know-how as to how to do each aspect of it. So how do we make it easier for everyone? So without the equipment, we can do those. So first, I will make the example how we perform long-term time-lapse imaging and tracking, and what did we learn from that. So this figure, I took it from a review that's written by Tim Schroeder 5 years ago. Essentially is to compare and contrast what different aspects of the biology that different techniques are capturing. Most of us are very familiar with the population average. Imagine that this bars or the 3 bars represents, for example, gene expression, and we're measuring this gene expression on a population average. That's the kind of bars we get. But in the past several years, more and more, the single-cell omics technology, for example, the single-cell RNA-seq, that everyone essentially studying our favorite cell types now is discovering, oh, our cell types that we've been studying for years is not pure. It's made up of a mixture of many different subsets or subclusters. And with the single-cell snapshot of gene expression, we can do a lot of analysis, for example, infer pseudo-time. Still, with the single cell snapshot, there are still certain aspects of biology that's not captured if you were to consider each cell that's its biology is different. For example, consider these 3 or 4 cells over time. Suppose that you can watch all these 4 cells over time, that they probably will go on to do different things. When do they divide? Do they give rise to 2 identical daughters or asymmetric, or different daughters, or do they give rise to many different progeny, or just a few? So the long-term live cell imaging tracking really enables you to look at each cell as its own individual, to probe the biology that's either transient and dynamic or rare. So in my lab particularly, we're obsessed by this kind of questions. If how does a cell type A becomes cell type B? Keeping in mind that A and B, they share identical DNA sequences, and how does A becomes B? That's usually driven by transcription factors, chemical signalings, or physical signalings, or the cell signaling. So to answer this question, one of the model systems we use is actually the Yamanaka reprogramming system driven by the transcription factors OCT4, SOX2, KILA4, and MYC. So in this movie, what you see is the yellow arrow is tracking a hematopoietic progenitor. Specifically, it's a granulocyte monocyte progenitor, or short for GMPs. In the beginning of this movie, it is colorless because it's not made to express any of the pluripotency reporter, which is OCT4 GMP. And occasionally, from time to time, you're going to see some of these cells would temporarily or transiently display the red fluorescence. That's because these cells were made to express the G1 cell cycle phase reporter. So when you see them being red, that means these cells are currently in the G1 phase. And the rest of the biology, I'll just fast forward so that you can see at the end or toward the later, around between 3 to 4 days, these GMPs that isolated from adult mouse now is beginning to express the pluripotency reporter OCT4. So now we can image this these cells one by one. We get to ask the question, how is this single GMP different from the rest of the hundreds of thousands of GMPs that are similarly expressing these same factors, yet they're not adopting a different cell fate? And because of these, we can map one mitosis to the next mitosis, and we can map the duration of how long the cells were expressing the G1 phase reporter. We can map out precisely how long each cell cycle lasts. And you can read by these timestamps here, these cell cycle, the first division cell cycle usually lasts somewhere between 6 to 8 hours. And let me remind you, the 6 to 8 hours of cell cycle is associated with a normal GMP state, because at this time, the virally transduced Yamanaka factors are not yet being expressed in large quantity to make any difference yet. So because of this rather unusually short or fast cell cycle that's associated with all these reprogrammed lineages, we started asking this question: what role does cell cycle speed or cell cycle length play in cell fate regulation? So because we saw that the short cell cycle is associated with essentially all the reprogramming behavior that we could image, we tested whether this cell cycle behavior is more generalizable, for example, in other somatic cell types. In this experiment, we just labeled or loaded a fibroblast culture with CFSE, which is a really generic protein dye. And every time these labeled cells divide, the fluorescent dye gets evenly distributed to its daughter cells. So you can see that the brightest fluorescence is just directly post-label, and then we let these labeled cells to continue to divide and program for 2 more days, and now you can see that dye gets quenched and gets diluted more. However, this one sharp peak now becomes more stretched, and now this dimmer population would represent the fastest dividing subset. And then we can sort from the same population, the intermediate cell population and slowly dividing cells. And now we get to ask which one of these cells can reprogram better. The answer is black and white, different. Essentially, all the reprogramming activity is confined within this perhaps 2 to 5% of subset of cells that are displaying about 8 hours per cycle division rate. So this is the kind of question that lead to me to establish my research program, is really to address how does cell cycle speed modulate cell fate regulation. Again, coming back to the question, what kind of role does cell cycle speed play in converting cell type A into cell type B? For most of the people here, perhaps you don't really care about the IPS, then what would be another relevant cell type? For example, the first additional the cell type B that we wanted to investigate is, how about oncogenes? How does oncogene convert a normal somatic cell into a malignant one? So to address this question, we decided also, what oncogene we want to study. So years ago, Dan Tennant's group published this really intriguing observation that for this oncogene MLL-AF9 to transform this particular progenitor stage, GMP stage, the same cell we have been imaging, seems to be particularly important for transformation. So this is, we're all familiar with this linear pathway. Stem cells become stepwise more committed to become GMPs and then give rise to more mature differentiated cells. In their experiment, if they ablate a GMP stage by deleting the CBP alpha, they completely block the development of leukemia. However, this leukemia development can be rescued even in the presence of CBP ablation if you treat the mice with additional cytokines such as interleukin-3. So these experiments suggested to us that this GMPs that we're very familiar with perhaps is required for ML-AF9 driven oncogene, driven malignant transformation. So to address the question then, does the cell cycle speed play any role in ML-AF9 driven leukemic transformation? We want to minimize everything else that we can think of in terms of malignant transformation. So to do that, we created a knocking allele of DOX-inducible ML-AF9 transgene and cross it with the ubiquitously expressed RTTA. So when these two alleles are together, you can give the mice or give the cells doxycycline, all the cells would be made to express ML-AF9. We know that all cells are expressing this oncogene because in the same vector, we put in the IRES and then followed by the delta NGFR. So with this marker is expressed on the cell surface that can be picked up by a fluorescently labeled antibody. We confirmed that these mice develop AML in genotype-specific manner and DOX-dependent manner. Now we get to image or ask the question, now can we image the leukemic transformation or de novo transformation with live cell imaging? So what you're going to see here are the primary GMPs before doxycycline induction that you're watching them. I think we're watching them, I forgot exactly what, but this should be at minutes of resolution. And you can see once the cells are transformed, they're going to start expressing this green fluorescence. In this case, the GFP is under the control of the MIS1 promoter, which is known to be activated or reactivated upon transformation. So I want to draw your attention, for example, this crazy cell that's really jumping or migrating around really rapidly seems to be associated with the transformation events. So in these kind of setup, now we enabled the tracking or imaging of de novo transformation from a normal granulocyte macrophage progenitor into a transformed one. Okay, so what I skipped through, didn't tell you, is that this GMP cycling behavior is very heterogeneous and dynamic. We know that the GMP population defined by the lineage negative, the conventional GMP phenotypic definition can be further fractionated, for example, into the bipotential neutrophilic versus monocytic, or the more defined monocytic versus neutrophilic. Even within the 24-hour or 1 to 2 days of in vitro culture, these subsets of progenitors, their cell cycle behavior is very different. These double negative or bipotential progenitors, they are still undergoing further cell cycle acceleration, while the monocytic progenitor, they are reaching or they are at the peak of their proliferation or cell cycle speed. The long story short is that, yes, again, in another cell type A becoming cell type B system, we're seeing that transformation driven by this oncogene at least is initiating from the already fast cycling cells. This oncogene is not responsible for converting a slow cycling cell into a fast cycling one. It is just working on top of the already rapid cell cycle. So again, this is the figure from the review written by Tim Schroeder. It's a scary plot. Essentially, this is the take-home message that he wants to say, and I absolutely agree, is that every long-term single-cell quantification experiment or project requires a lot of optimization, depending on the kind of question that you want to address. So in these many blue parts here present a really high threshold as to you may not consider long-term imaging as your option for addressing your question. So we want to change that. Specifically in terms of long-term imaging and in terms of determining the cell cycle length or cell cycle speed, what can we do? I told you briefly that even among the GMPs, the subsets of GMPs, their cell cycle rate or cell cycle speed is very different and dynamic depending on when you're measuring them. And this cell cycle speed also changes during differentiation. The one important thing is the way that we have been measuring the cell cycle speed is by watching them divide once and divide twice and divide again. So to calculate the time interval between two consecutive divisions. And in other words, it is always somewhat retrospective. We really wanted to come up with a way that can immediately, on the fly, on the go, can we tell how fast the cell cycle is running at. So to do this, we put together a mathematical modeling in collaboration. This part of the work is in collaboration with Dr. Haoyuan Kui from UWash. Essentially, the mathematical model suggests that molecules of differential half-life would be differentially susceptible to the cell cycle length or cell cycle speed. Stable molecules would be diluted more or reduced to a larger extent as compared to the labile or short-lived molecules. So the way we did it is to connect a color-changing protein called fluorescent timer with the H2B that we created the fusion. And this timer protein's a unique feature is that when it's newly synthesized, it's a blue fluorescent protein. And after a couple hours, it irreversibly convert to a stable, much longer lived red fluorescence. And for all today's discussion, all you need to remember is that faster cycling cells, by this mathematical modeling, the faster cycling cells would be blue versus the slow cycling cells would be red. So if this strategy works, then you don't necessarily need to wait at the microscope to wait for the cells to divide once, divide twice to calculate their cell cycle speed. You just need to measure their fluorescence ratio for how red or how blue they are. This paper is published last year, so I won't go into the details, but just to show you how these images would look. It doesn't really matter what tissue this is, but you can see that on the tissue section, this is a frozen tissue section, that the cells would show up with a different shade anywhere between blue and red, and anywhere in between. As I said, the faster dividing cells are bluer, and you can see this this mouse is posted by a half-an-hour EDU, and this blue cell region is co-localizing with those that are positive for EDU. As you would expect, faster dividing subset or populations would have a higher percentage of those cells at any given time to be in S phase. But I also want to point out there are many more blue cells and the EDU positive cells, because even these cells are all dividing more rapidly. Not all the fast dividing cells happen to be in S phase when the cells are paused, when the mice are paused. So in another view, we can also see that faster cycling cells appear bluer. This is a very simple cell line. BAF3 cells, which proliferates depend on how much interleukin-3 you're feeding the cells. When we're growing the cells in the full concentration of IL-3, these cells divide very rapidly and a lot of them are blue. And we can reduce or cut down the IL-3 concentration. And when we completely wash out the IL-3, you can see then the whole population essentially shifts to the red, and they're slower. And the way to, the one way we can also depict the cell cycle speed is by measuring or reporting this normalized blue to red ratio, being 1 being completely blue and 0 being completely red, and the cells on this side would be the faster ones. So you can see that, with the increasing concentration of IL-3, the cells proliferate faster and faster. And this fluorescent reporter now is, again, made into a transgenic reporter mouse. And this is the image that I put in a cover slide. This is a cross-section of the reporter mouse's bone marrow. I'm hoping that multiple of you, or many of you, would be interested in some parts of the marrow that you can see. It's a mixture, again, with different colored cells. We yet need to further utilize the potential of what this reporter mouse can enable. And if you're interested in collaboration or using these mice for the lineages that you're interested in, please contact me. So in this reporter that we know, following a calibration, this reporter can immediately tell you how fast your cell cycle is running. For example, this TER11 positive erythroid progenitors, by this method that we're measuring, their cell cycle length, or the subset that we're measuring there, it's shockingly only about 4.3 hours per cycle. And the GMPs that we were measuring there, it's somewhere between 10 to 12 hours per cycle. And here I'm only showing you, for example, the GMPs and the lineage negative KIT-SCA positive, the HSPC enriched populations, and the whole bone marrow being heterogeneous. You can see it spans the whole spectrum of the blue to red ratio. So the nice thing that we like about this reporter is not only that you can see how fast your cells are dividing, now it is compatible with the conventional flow cytometry method that's already you're using it in your lab. Now we can pick the cells out, fractionate them however you like, based on their fluorescent blue to red ratio. So for example, these are the same subsets of GMPs we know that are already dividing fast, and we subfractionate them into the monocytic versus neutrophilic poised or committed population. And now we can see these more committed ones, as we were tracking them by imaging, they appear to be even faster than the bipotential ones, which is yet to undergo further acceleration. And then we also tested further in the erythroid progenitors. For example, we know that erythroid progenitors would undergo regeneration if we treat the animals with phenylhydrazine to induce hemolytic anemia. And I believe this is done in collaboration with Shilpa. I know she's here. I think that the population we're projecting here is the orthochromatic progenitors. You can see that in the black lines are the baseline PBS treated animals, that in the bone marrow, there's a lot of the erythroid progenitors that are very blue, indicating they're cycling fast, and on the resting state in the spleen, that's not so. But once this mice were treated with phenylhydrazine, yeah, you can see that there's a further blue shift. And in this reporter, that would indicate further cell cycle acceleration in both the marrow and spleen. That's underlying or coinciding with the erythroid cell regeneration.
So this is Diane, just a question there. Is
Yes.
that, do you know the mechanism? I mean, is it because they've now got more EPO receptors on them? I mean,
Great question. I know nothing about erythroid progenitors or erythroid biology. All I know is that though we can capture this color shift in this reporter. So for anyone who is interested in this kind of question, I'm more than happy to talk to you more about how to use it. But I'm sorry, Diane, I don't really know anything about erythroid progenitors other than they appear to be accelerating.
so Stefano Ravella has a question. Stefano, do you want to unmute and ask?
Sure. I mean, I guess it probably is similar to the previous question, so maybe we can talk later,
Sure.
especially if this model is available. My question was more about the different populations, since you're showing here, you know, the typical CD44 TER119 profile. I was wondering if you were able to look at single population and see a better, you know, definition of the cell cycle in each population. But if you don't know right now, we can talk later. It's okay.
Yep, let's talk later because really I'm quite ignorant about the erythroid biology, but yeah, that's the purpose today. I'm looking for anyone who is interested in this aspect of biology.
Hal Broxmeyer also chimes in and
Okay.
says, for another time, it would be interesting to be able to check for transferrin receptor expression on the cells. So there's a bunch of ideas of how to actually see maybe what's mediating this increased cell cycle. Just as an aside, as a really iron focused person, just giving them more iron, does that increase their cell cycle speed, like faster?
This again, I have no idea. I mean, I know Daniel is here, that he does know quite a bit about erythroid progenitors, and I know they have a paper in revision using this system. So, but unfortunately, I cannot answer really any erythroid specific question, but I'm more than happy to participate in the conversation. Okay.
So if I can maybe make a point, or you know, provide some idea, I would expect not necessarily in a steady state condition, as hepcidin knockout mice, they don't have a lot of iron. They don't really have more erythropoiesis, but they have more hemoglobin per red cells. So maybe they will get bigger, but more dense of hemoglobin, but not necessarily making more red cells.
But it's about cell cycle speed.
Yeah, but
just to add to that, I in my past research, I mean, I've been looking at also rescuing slow cycling cells with the addition of iron, mainly by using an iron carrier that is cell permeable, bypassing CD71 signaling, which was FESIH. And in that case, we didn't see a faster S phase. There was a slight increase, but nothing significant. However, to answer your question, Stefano, we did look with actually using this great model that Dr. Guo has. We did look at erythroblasts such as Eri A, Eri B, Eri C, and we're able to see the differences of actually cycling upon EPO stimulation. And we can definitely discuss that a little bit more.
Yeah, that would be great to know. That would be a really nice, probably nice piece of information. Thank you.
Well,
Excellent.
thank you for questions and comments, everyone.
All right, so I'll move on then. Okay. So again, to continue to drive this message home that this reporter is made into a mouse, you can cross it with whatever you like and whatever cell population you're interested in. And for example, we're interested in the leukemic transformation. We can see and this again, this is measuring the blue to red ratio in the GMPs expressing inducible MLAF9 oncogene, or not AF9 oncogene. This actually is the MLENL oncogene. So at the disease initiation, we can see a small number of very blue cells already there, but at late stage, this is entirely driven or taken over by the leukemic GMPs. So it has the flexibility that you can, because it's a DOX-inducible model, you can choose your TTA or RTTA and restrict the reporter in the cell types that you're interested in.
So I have, sorry to keep interrupting, just one more question on that.
Yes.
Is the win out of the blue cell cycle change out competing for nutrients? I mean, we talked about iron, but could these guys be winning for nutrition, and that's why that population gets more dominant over time?
It's a possibility, but in this case, I don't think it's even necessarily need to compete for anything, just by the sheer cell cycle rate being faster, given that this is probably different by 3 weeks, these two states. In 3 weeks, if we calculate that these cells are dividing, let's say, 8 hours per cycle, this is maybe 24 hours per cycle. We can calculate really how much more this population is going to expand in relative to these cells. And just by a sheer number game, this could be explaining how transformation is evolving. But we haven't done that yet. We haven't really done the calculation in terms of cell number, because it's also offset by, as you said, nutrient competition or cell death, or immune checkpoint, all those things. But now we do have this measure in vivo that you could do.
Great, thanks.
So after COVID hit, of course, we wanted to see, for example, is there anything else that we could do to look at in the tissues? We then looked at the embryonic lung. Then you can see this very nicely organized the tissue structure. Again, I know nothing about the lung. I just am happy to see the color distribution, that it's revealing something that someone might be interested to study. However, what I want to use this slide is to tell you that although this reporter is great in helping us to address some of the questions, the student who worked with this model, now graduated, she would complain that because it's a timer, it's constantly changing color. Forget about having a coffee break when you're working with this. Just go, go, go. Otherwise, it's going to change the color. So we saw this really firsthand by this observation. That these colors were acquired immediately after the tissues were sectioned, frozen section. Because this timer protein will undergo chemical, when you fix it, the color undergoes permanent blue to red conversion. So you lose all the time resolved information. So on the frozen tissue section, even if you store it in the freezers over 6 months or demanded by this lockdown, what we found, unfortunately, is if you store these sections for extended time, all these timer protein would eventually convert to become red. So because of these limitations, the inability to undergo chemical fixation, that's motivating us to make the cell cycle speed reporter version 2.0. In this case, we're going to remake the design is to have everything that the original reporter that already has, so live cell without anything further labeling, all those will be contained. But in addition to that, we will put in a membrane-directed destabilized halo. The reason for that is because then we have a very bright fluorescence that's limited to the cell membrane that can be chemically fixed. And the instability is going to be induced on 2 levels. One is the destabilization protein domain. 2 is destabilizing RNA module. And this right now is only on the paper. We're yet to make this. So this is not yet available, but for anyone who's interested in the H2BFT part, yes, it's anyone is welcome to use it. Yeah, so that ends the part that the first part that I would talk to you about, reading the cell cycle speed by this fluorescence reporter. And this the work that I showed you so far were led by two very talented graduate students who both graduated in the past year during the pandemic. But as I said earlier today, that my mission today is not only to tell you what my lab does, but really to tell you what the imaging core of the Yale CCEH would like to enable. So the next, for the next 5 to 10 minutes, I would want to show you the work by my colleague, Jörg Björsdorf, in driving the sort of resolution aspect. So usually that resolution to cross the light diffraction limit, we are talking about super resolution, and this again requires very specialized equipment and technical know-how. So for every one of us who are familiar with light microscopy, what can we do? So what Jörg and his lab did is to devise or further devise this expansion microscopy. So if you don't have the super resolution microscope, then let's make your cells or specimen bigger. So this part of the protocol, again, is published in Nature Communication last year. You can find the citation in the subsequent slides. So this procedure requires stepwise expansion. And after 2 rounds of expansion, linear expansion now is undergoing about 6 to 20 fold of expansion. So for the 3D expansion, you can calculate that's many thousands of expansion. And with that expansion, I'll show you some of, this is the publication citation. So we're mostly familiar with the hazy, images without expansion under confocal microscopy. After 1 round of expansion to go 4-fold, you're about to starting to see the subcellular organelles. And with the second round of expansion, now some of these subcellular structure is much better resolved. And we can see now in the zoom in, this boxed region, now you can clearly see the cristae from mitochondria or the Golgi stacks. And and also you are going to emphasize that this image, all these images are acquired on the standard confocal microscope. So you can do this yourself. And side by side, this is what's acquired by the expansion microscopy. This is taken from a published figure that's acquired by electron microscopy. So some of these images you can really see the quality is quite comparable. And because this is acquired on the confocal microscope, you can imagine that we can acquire 3D image stacks. You can see that the fibrils in the nucleolus, and some of these really bright dots are the nuclear pore complex, and some of these Golgi stacks, when we're going through the cell, now they are beautifully resolved. And this expansion microscopy technique is compatible with most immunofluorescent antibody staining procedures. And you can see some of these images. You can see the mitotracker staining the inside of the mitochondria, and the TOM20 staining the mitochondria outer membrane, and this ER marker SEC61. So overall, the it's called expansion micro PEM XM. Can enable the samples to be expanded in linear dimension about 16 to 20 fold. It retains the protein information, but you can acquire the whole cell image. Really what it enables is the correlation light and electron microscopy like images with your regular confocal microscope. What I want to say on behalf of our imaging core is that this technique is currently being optimized for hematopoietic cells, because really, as the blood cells that's near and dear to our heart, for imaging, it poses many unique challenges. but we are working on to reduce or eliminate such limitations. And this is the group, as I said, Jörg Björsdorf's lab in innovating and driving this technology. This is my last slide. Again, for the purpose of today's talk, I'm really presenting on behalf of the Yale CCEH Imaging Core with the mission to really make the imaging technique more widely accessible to all of you, be it the long-term live cell imaging and tracking, or be it, calculating time-resolved parameters based on one snapshot measurement of fluorescence, or making your samples bigger so that ultrastructural level information can be reviewed by conventional microscopy setup. I will stop here and let's see whether we have any questions or discussions to have.
I hate to dominate the conversation, but have you looked at, can you do other eukaryote cells like CERVCA?
For which part?
For that microscopy pan expansion?
I think that the images that I showed are all HeLa cells or 293T, this kind of really big flat cells adhering, but I don't see why their technique cannot be used. But again, I'm speaking on behalf of Jörg. It'll be better for him to answer the question, but I don't see why it can't.
Thanks. Okay, any other questions?
I have a question. I'm more a generic, very generic question. So it looks really like the idea of a way to look at the relationship, somehow, relationship between proliferation and also differentiation. I wonder if you also look at more in particular, let's say hematopoietic stem cells or other system where generally fast proliferation is associated with differentiation or loss of stemness. That I'm just curious if you have any data in that direction or can indicate, teach us something about it.
Okay, so that's exactly what my lab is interested in. One of the systems that we use quite heavily is the Yamanaka reprogramming system, is particularly starting from the hematopoietic progenitors. Recently, we realized that for the cells that are undergoing this very unusual rapid division or cell cycle, one of the things they remodel most is the actin cytoskeleton or the actomyosin system. And what does that have to do with the chromatin or epigenetic reprogramming? It turns out that the cytoskeleton or the mechanotransduction system is ultimately feeding into the genome reprogramming. And for the pluripotency to initiate, this mechano or somatic mechano transduction system need to be eliminated or reduced to very, very low level. So mechanistically, that's our part of the insight. Whether this is generally applicable to all somatic cell types, we don't know. But we're also very interested in, for example, the how cell cycle dormancy or length underlies, let's say, hematopoietic stem cell fate determination or chemoresistance in the leukemic setting. These are some of the questions that we're very, very interested in. But with Anna, or the first author who made the reporter mouse graduating, and with the pandemic, everything, we yet to need to make those happen.
Thank you.
Okay. Any other questions?
I see a couple things in the chat. Should I?
I think those have been asked and answered. So I think we're okay there. Thank you for noticing that.
Mm-hmm.
If not, I'm gonna ask you. Oh, go ahead.
Oh, sorry, just if you want to work on we have a different mouse model, so disease mouse model. Maybe we can talk later. Is there a way eventually to collaborate? I would be very interested in looking at how these different defects affect erythropoiesis. And I wonder if it's something you're open to discuss.
Absolutely. Again, I'm speaking really on behalf of the imaging core. Anything we can do to help. That's what NIH is paying us to do.
Okay.
But for anyone who's listening here, all these reporter constructs are available in viral forms. So without shipping you any cells, mouse, if you just want a quick and dirty way to test to see whether it works in your system, we've deposited the plasmids in Adagene. You can get them.
So it means we need to do the virus and do bone marrow transplant?
Or if your biology can happen in vitro, that's an option too. Yes.
That's probably more challenging, but you're saying that if we do bone marrow transplant with this vector, then we can image the bone marrow cells.
Absolutely, we've done that. Yes.
Okay, got it. Thank you.
Mm-hmm.
Great. Any other questions? And just a reminder, if you find that this might be a useful tool for you, pilot and feasibility money might be an option for those of you who are trying to develop this for a research program from the Centers of Excellence in Hematology, where you can get some funding support to start these sort of initial ideas. And that's at the same website that I showed you at the beginning. I want to thank everybody for their participation, and especially I'm stunned by the microscopy. That's pretty great. So thanks for that. It looks like we all need to go look at our favorite hematopoietic cell with that kind of microscopy to inform really about timing on differentiation. I think it would be remarkable.
Yeah, that's what we're thinking too. So thank you, Diane, for mentioning the CCEH, the U24 pilot grant, right? So if any of you find any of these tools might be helpful for your kind of questions, let me know. We're more than happy to provide in any way we can to help you to advance your question, your research.
Yeah, and if you need me to connect you to someone, just reach out, send out an email, and any of us can try and assist in that. So make these connections for you. Again, thanks everyone for your participation. And our next talk will be held on June 2nd by Vijay Shankaran from Harvard and Boston Children's Hospital. So hope to see you again then, and encourage you to continue these discussions through emails or your own independent Zooms to help strengthen our hematology research piece. So thanks everybody. Take care.